My guest today is Professor Rob Lustig, neuroendocrinologist and Professor Emeritus from the University of California, San Francisco. He's a previous guest on this podcast and world-renowned for his books Fat Chance, which really showed us so clearly how it's sugar in its various forms, including fructose, that is driving health problems, not fat. And metabolical, that went deeply into unraveling how sugar, fructose, and ultra-processed foods degenerate health. And also a book that I personally love, The Hacking of the American Mind, which explained how addicted we become through this process of refined food. But now, and not in any book, he proposes that there is one messed up underlying process that almost all contributors to cognitive decline compound. And that's what he's here to talk about today.
So, I'm honored to have you back, Rob, doctor and professor.
Not your doctor, it's okay.
Endocrinologist, and one of the few who really studied nutrition. And of course, all of us know of your amazing work, going back to Fat Chance and also Metabolical, which I thought was a masterpiece. And many people will sort of pigeonhole you into the area of kind of anti-sugar and ultra-processed foods driving, so many of our problems. But I'm here to learn more about a very interesting hypothesis you have, not only on dementia, but of course that's been a focus of my work for a while, which is that many of the things that drive us into cognitive decline have underneath them a common process that's going wrong.
Right.
What's your theory, Rob?
Well, so there are so many different things that can affect the brain. Everything from food to air pollution to pesticides to stress to chemicals. There's so many different things that have been associated with the development of dementia. The question is, how do they all dovetail together? Is there a common pathway, and is there a common intermediate that explains all of these? And after looking at this question vigorously and carefully over the last, I would say, 2 years, I now think I have an answer for you. It's three words, 3 letters, ROSs, reactive oxygen species, oxidative stress. Now, this is not new. There are plenty of studies in the literature. There are plenty of review papers in, the medical literature that argue for this. This is not a revelation. The question is this the common antecedent? When you look at the various things that people have identified as being causative for dementia, ROSs always pop up, but then there are a whole bunch of other things. And, for instance, telomere shortening, cortisol, etc. But actually, ROSs drive all of those. So those actually may be downstream of the ROSs. And so ROSs may be primary. And so that's the idea here. Now, what got me thinking about this in the first place? So sometime last year, there was a paper that appeared in the journal Cell Metabolism from the Buck Institute, very close to where I am in San Francisco. And what it showed is something that I did not know, something really, really different and new and interesting. And that is that there are glycogen molecules in Alzheimer's neurons. Now, there's no reason.
And for those listening, glycogen is like a a storage sugar. We go from glucose, and the body stores in the liver and muscles glycogen. And you're saying there's glycogen molecules in the neurons of people with,
Right.
yeah.
Now, we've known there's glycogen in the brain for, quite a while, for I would say 3 decades. But every time that someone stained for glycogen previously, it was in astrocytes. Astrocytes are the cells that nourish the neurons. They basically provide backup for neurons, and they basically feed neurons what are needed. Neurons are so friggin' busy. They have so many mitochondria. They have to generate so much energy in the form of ATP, adenosine triphosphate, that they don't have the capability of storing energy. They have to burn it straight away to get carbon dioxide and ATP. Glucose comes in, carbon dioxide and ATP come out. There's no place to store it. And so no one had ever noticed glycogen in neurons before. Like I said, glycogen in astrocytes, sure, small amount, but glycogen in neurons, never seen before. But this paper very specifically said there's glycogen in neurons. And I'm going, what the hell? If neurons are so metabolically busy, which they are, I mean, your brain is 2% of your body weight, but it uses 20% of the energy in your, that you consume. Okay, so it's 10 times greater in terms of energy utilization than any other part of your body, including muscle, including the heart. If brain is so busy and has so many mitochondria, how in the world does it store energy? Why would it store energy? What's the purpose of that? And then it came to me. It's not a purpose. It's a mistake. It's not supposed to do that. Um, so I've wrote a couple of papers back in 2022 through 2024 about a different phenomenon called obesogens. Obesogens. These are chemicals in the environment that drive adiposity. And colleagues of mine, Jerry Heindel, Barbara Corky, Sarah Howard, and I wrote a paper in January of 2024 in the International Journal of Obesity called The Redox Model of Obesity, basically arguing that what was happening was that you have mitochondrial dysfunction, and that prevents glucose from being burned all the way to ATP and carbon dioxide in the liver and in adipose tissue cells. And so the excess that's not getting burned has to be diverted and stored. Now, in the liver and in the adipose tissue, that storage occurs as fat. Now, in the brain, you can't do that. It doesn't store as fat. What is it being stored at? It's being stored at as glycogen. So basically what this told me, the fact that there's glycogen in Alzheimer's neurons, said that this must be a mitochondrial problem, that Alzheimer's must start with a defect in energy usage and utilization. Now, again, this is not rocket science. This is not new. This has been around for a long time. The question is why? What happened? How did that occur? And that's where this whole obesogen thing actually sort of came to bear. And if there's something new and novel in the story, this is it. So when you look at all the different things that cause Alzheimer's, and there's boatloads, okay? And of course, food being, at the top of the food chain here, being, the most important. Mitochondria have to burn energy to ATP. That's their job. They have to take the glucose, the glucose gets converted to pyruvate by an enzyme, a process called glycolysis, and then the pyruvate enters the mitochondria, gets converted to acetyl-CoA, enters the Krebs cycle. Krebs cycle turns, and what you get out of it is carbon dioxide and ATP. And that ATP then is utilized by the neuron to do all of the things that neurons do. Most important, of course, is neurotransmission, firing. And it
Hmm.
turns out firing is very, very energy expensive. And that's why the brain has more mitochondria and uses more energy than anything else, because in order to fire, you have to have all these ion fluxes, and then you have to pump the ions back out again. And that costs a lot of energy. So neurons are very energy intensive.
Now,
And so,
just to back up here a little bit, one of the great ironies that is occurring in the world of dementia and Alzheimer's is that the consumption of too much sugar, and you've also highlighted fructose and ultra processed foods,
right,
which should in effect be energizing the brain. The work of Professor Stephen Cunnane
right.
showed that the glucose isn't working. And I think that's what you're
That's
saying with
right.
glycogen. If you've got glycogen in neurons, it shouldn't be there. So it's,
That's right.
and then what happens in dementia, and anyone who cares for people with dementia will know, is that people are experiencing brain fog. And what is their system telling them to do about it? And it says, eat sugar. I need fuel. I'm lacking fuel. I want
Well,
sugar.
indeed, I need fuel. The question is, do you need sugar?
Exactly.
They're not the same.
Mm.
They're not the same. And that's part of the issue here. Now, my work previous to this, and other people's work as well. So I mean, certainly not alone in this. Shows that uh, the molecule fructose, this sweet molecule in sugar, the molecule we seek, the molecule we crave, the molecule that's addictive, is a dose-dependent mitochondrial toxin. It inhibits three enzymes in the mitochondria that prevent that burning all the way to ATP and carbon dioxide. It inhibits um, uh, 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 AMP kinase, which is the fuel gauge on the liver cell. It inhibits ACAD-L, acyl-CoA dehydrogenase long chain, which is necessary to cleave the fatty acids into two carbon fragments so that they can be burned for energy. And it inhibits CPT1, carnitine palmitoyl transferase 1, which is the shuttle mechanism to get the fatty acids into the mitochondria for burning in the first place. The sum total of which means that the molecule fructose inhibits burning. So you've got all of this glucose coming in from the bloodstream into the brain, and the mitochondria are dysfunctional. They're not working. So what happens to the glucose? It gets converted to glycogen.
Hmm.
This is the the nugget of truth. This is sort of where we are here. Is you've got basically a mitochondrial dysfunction with a diversion to a storage form. Now, in the liver, it's fat, but in the brain, it's glycogen. And the neuron doesn't want to store glycogen. That's the key. It doesn't want to, but it has to because the mitochondria are not able to do their job. So the neuron is starved for ATP. You have a defect in ATP generation. So the way to think about this ROS business, how do you pull this ROS thing into it? Okay. Now, mitochondria make ROSs normally, even when they're working perfectly. One molecule of glucose generates 11 ROSs.
And by the way, these reactive oxygen species, I mean, you could think of them a bit like the exhaust fumes of reacting glucose with oxygen. They're
Yeah,
always produced. Yeah.
they're always produced. You can't not produce them. They're baked into the cake, as it were. You can't burn glucose without generating ROSs. Period. Every molecule of glucose generates 11 ROSs, whether you like it or not. Now, those ROSs are not just toxic molecules. They can be, of course, in high dose. They can do oxidative damage. They can cause protein denaturation. They can cause lipid peroxidation. Ultimately, they can cause cell death. But in low dose, in low dose, they're signaling molecules. They're telling the cell what's going on energy-wise. So when the ROSs build up, what they're saying to the cell is, hey, I'm busy here. I got too much crap coming down the pike. I can't deal with it all. Divert, divert. And so what the ROSs are doing is they're changing the phosphorylation status of three enzymes, one of them being AMP kinase. And AMP kinase is necessary to build new mitochondria. So you have a defect in being able to process energy through mitochondria, and you have a defect in being able to build new mitochondria. So you've got an ATP generation problem that the ROSs are actually instigating. And the only way to not do this, the only way to manage this problem is you have to get rid of the ROSs. Now, what gets rid of the ROSs? The antioxidants. They're in the peroxisomes. Okay, so glutathione being the big one, which of course is why you are so interested in the glutathione index, and correctly so. Okay, and vitamin E, and and several other bioactive molecules, which we'll leave for the moment. But there are a whole bunch of antioxidants that are necessary to get rid of the ROSs. And the ROSs then are like throughput. So the way to think about this is like this, an analogy that will make sense to the audience. You have an old style 19th century factory before electricity. Okay. Now that factory needs energy to run the furnaces. So where's the energy come from? Well, it came from coal. And the coal would be shipped in on coal cars on railroad tracks that would come straight into the factory. And then there are these stokers, guys with big shovels that would basically unload the coal from the coal cars and then basically straight into the furnace. And then the coal cars would leave in order to make room for other coal cars to come in. And so there was an equilibrium between how fast the coal cars showed up at the factory and how fast the stokers could unload and stoke the furnace. And when everything was optimized and you had the coal cars coming in at the same rate that the stokers could stoke the furnace, then the factory worked at peak capacity. So far, so good?
Mm-hmm.
All right. Now, what if those coal cars started coming a little faster and a little faster and a little faster? Now, those stokers are going to have to work a little harder and a little harder and a little harder, and they're going to be stoking that furnace in order to basically just get the stuff unloaded and into the furnace. But you know what? You work that hard, and maybe one stoker, pulls a back muscle. Another one breaks a leg. Another one, gets burned. Or, they start falling off the line because of injury. And now those coal cars are still coming so fast. And now you don't even have the stokers to be able to unload. And so what's going to happen is those coal cars are going to start building up, and ultimately those stokers are going to fall off and fall off and fall off until finally there are no stokers to unload the coal, and the coal cars basically sitting on the tracks, and nobody can take them away, and ultimately the factory shuts down.
And that's the glycogen. So now you're storing glycogen in neurons and you have no energy and you're generating reactive oxygen species.
Exactly right. So you're basically screwing your mitochondria. You've got the energy, you're just not able to utilize it.
Now
And in the process, you have to store this glycogen molecule, which you shouldn't have to. And that becomes a problem in a minute, which we'll talk about further down the line. Okay, so just hold that thought for a minute, that the glycogen has to be dealt with, in a different way. Okay. So you have a problem in ATP generation. And it's because the ROSs are feeding back and diverting the energy away from mitochondria and toward storage. And this is what happens with obesogens in the liver. And so I believe this is what's happening with dementiagens, if you will, in the brain. And it doesn't matter which dementiagen you choose, they all generate ROSs. Pick your dementia causing agent. It's an ROS generator. And I made a little table of obesogens versus dementiagens, and they are virtually superimposable. Everything that causes obesity also causes Alzheimer's.
There's three conversations that I had not so long ago that make me very excited about your hypothesis. The first was with Dr. Tom Levy, and he basically said all nutrition boils down to oxidation, antioxidation. We are a oxygen-based life form processing glucose for energy. It generates reactive oxygen species. Ultimately, we die. So that was one thing. You can really say that's central. And then the
No, I
other
think
thing
it's
that's fascinated me, and I've never fully got an answer, although all the experts I've spoken to think it's probably true, that basically in all plants, well, not all plants generate fructose, and animals, we generate, we burn glucose.
right.
And when a plant makes fructose, it
Mhm.
also generates ascorbic acid
Yes.
from fructose. It's really
Yeah.
very similar. Same with glucose. Animals make ascorbic acid. We don't. We've lost that ability, but nearly
Right.
all animals do. So it's a little bit like there's this funnel where we're making the fuel, glucose or fructose, which will be burned to create ATP, which will generate oxidants, and you've got to have an antioxidant. So my proposal here, which I haven't really found an evolutionary biologist who can stamp this very clearly, is that we could not have an oxygen-based life form burning glucose or fructose without also creating the antioxidant. And
Well, we have
that's how
to.
I got into this through Linus Pauling. And, why did he focus 38 years of his life on ascorbic acid metabolism? Very interesting.
Well, well, indeed, we have to have those antioxidants, and especially in the brain, because more, glucose metabolism occurs there than anywhere else. And like I said, those those mitochondria generate ROSs whether you like it or not. They're baked into the cake. But here's the problem. The ROSs come from within, from the mitochondria, but they also come from without. They come from all of these exposures that we now currently find ourselves in. So air pollution, ionizing radiation, microplastics, BPA, PBDEs, phthalates, neonicotinoids, fructose, all of these things that we never had throughout our entire evolutionary history that we are now exposed to. And every one of those is an ROS generator. And every one of those has been associated with dementia. And by the way, every one of those has also been associated with obesity. This is what I mean by the obesogens are also the dementiagens, because you have basically double ROS. You have the ROSs that your cells make because that's life. And you also have the ROSs that you're exposed to, which is our new life. So we have double ROSs we have to dispose of. And we don't have the antioxidant capacity to be able to manage it, in part because ultra processed food is antioxidant poor. Because most antioxidants don't taste very good. They're bitter for the most part. I mean, they're not, shall we say, highly palatable. And also antioxidants, because they're antioxidants, spoil. They go rancid. Okay. A lot of them have cysteine groups, sulfhydryl groups. Okay. And sulfhydryl groups are very easily oxidized to, things that don't smell very good. And so it has behooved the food industry to get rid of the antioxidants in order to make ultra-processed food last on the shelf. So the more ultra-processed food you eat, the less antioxidants you are consuming, and therefore the less you'd be able to stave off this ROS onslaught that is occurring from both within, that is mitochondrial dysfunction, and from without, that is all of these exposures in the exposome that are associated with Alzheimer's. So this is step number one. Inadequate ATP generation due to mitochondrial dysfunction and ROS production. This is only step one. There are 4 steps.
What's step number 2?
Step 2, cortisol. Okay, glucocorticoid exposures, stress, okay, cortisol for whatever reason. And why? Because number one, we are stressed like never before. Chronic stress is now our, long-term companion. We used to have acute stresses. Now we have chronic stresses. And the thing is that what stress does by activating the sympathetic nervous system is it increases neural firing rate.
And by the way, just one point here is that we have been described as the stressed ape. Coming back to vitamin C, if you take a fish like a trout, which makes vitamin C, and a teleost or Oscar Amazonian fish, which does not, and you stress them, and by the way, don't ask me how they stress a fish, the ones that the ones that make vitamin C will produce much less cortisol. One of the functions of cortisol is to tell the liver to make vitamin C. So we can think vitamin C is almost like a hormone, a stress hormone. So we, because we do not make vitamin C, produce a lot more cortisol. We only have one, gun, so to speak, not the two, ascorbate and cortisol. So we, as a species, because we've lost vitamin C, are particularly stressed, particularly producing cortisol, and even more so if we watch the news and politics and emails, new apps. We are just a very, very stressed ape.
We very much are, and we're not doing a very good job of managing that stress, for sure. Point is, cortisol and stress in general increases neural firing rate throughout the brain. Well, I just told you that neurons, when they fire, they need a lot of energy in order to be able to reset. Okay, and action potential is very energy intensive. And so when you are stressed, you are generating a whole lot more action potentials than you are at baseline. So you have an increase in ATP utilization. So step 2 is increased ATP utilization. Step 1 was reduction in ATP generation. Step 2 is increased ATP utilization. Well, now you've got ATP depletion. Now you've got an energy crisis. So how are you going to feel?
Well,
how are you going to feel if there's not enough
ATP? In the UK, we say knackered. I don't think the word works so much in the US. And not a naked ape, said a kid in the exam, but a knackered ape.
Yeah, well, we sort of have it here too, but we're all pretty knackered.
Wired and tired. Yeah.
So tired, okay, brain fog, depression, okay, inability to focus, loss of attention, all of those, okay, and that might get you to the doctor. And if you told your doctor, this is what's going on with me, what would they do? They would probably give you an SSRI. They'd probably give you a serotonin reuptake inhibitor because they'd say, oh, you're depressed. Well, and in fact, you are depressed. But you're depressed not because of serotonin deficiency. You're depressed because of ATP deficiency. And the serotonin reuptake inhibitor is not going to do a damn thing about that. And when you look at the data on SSRIs in depression, they only help about 35% of the population. The other 65% are not helped. And I think it's because they're treating the wrong thing. And by the way, depression has an odds risk ratio of 2.85 in terms of developing dementia,
Mm.
because step one and step two. So now that leads us to step three. And step three has two parts, 3A and 3B. So let's do 3A first. You have a reduction in ATP. What's ATP used for inside neurons? What's ATP used for inside any cell? Okay. You have to fold proteins, protein folding. Okay. There's this phenomenon called the unfolded protein response, UPR. Okay. It's been associated with metabolic syndrome. It's been associated with dementia. Why? Because when you fold proteins, you have to bring different um, uh, amino acid structures closer together or farther apart that are against their standard energy capability. They have to basically defy entropy. And that requires ATP to be able to do, to bring two amino acids that are likely, that have like charges closer together. You have to basically, force it to do so. You have to basically, uh, put energy into the system to be able to fold proteins properly.
So this is like building. This is the building process.
Yes,
All the things
absolutely.
we need to build to stay alive and well.
Well, one of the things that has to build properly is this amyloid precursor peptide. And if you don't fold it properly, what do you get? You get amyloid beta. If you fold it properly, you get SAPP. But if you don't fold it properly, you get amyloid beta. And this is an ATP dependent process. So the lack of ATP, more A beta. Okay. And
Hmm.
that A beta is going to ultimately be reflected in the development of plaques. Okay. That's 3A. 3B. Remember that glycogen?
Hmm.
Well, glycogen is not supposed to be stored in neurons. There's no storage place for for glycogen. So you have to make one. Well, you know what makes it?
What?
Tau. Tau is the cage
Hmm.
for glycogen storage in neurons. Now, the way it works is the tau is basically supporting microtubules, which are basically helping move, stuff up and down the axon and, through the dendrites, getting stuff here, there, everywhere, all throughout the neuron, okay, through the microtubules. And the tau is the supportive structure for the microtubules. But when glycogen starts being manufactured, you have to basically develop a cage for it. Okay, and the tau comes off the microtubules. And how does it do that? It gets phosphorylated to something called phosphotau 217.
P-tau.
Well, p-tau 217 is a marker for Alzheimer's, right? So is
Hmm.
A beta. Okay, well, here's where the markers come from. They're coming from the ATP depletion. And so the phosphotau 217 is phosphorylated by an enzyme, okay, a kinase, right? Because that's what puts puts phosphates on proteins is a kinase. And you know what that kinase is called?
No.
Glycogen synthase kinase 3 beta. Why is a glycogen synthase kinase phosphorylating tau? What the hell do those two things have to do with each other?
ROSs.
No, 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 no. I mean, the tau is the cage. So
Mm.
the cell knows that the glycogen is unwelcome, needs a cage. And so at the same time it's making glycogen, it's making phosphotau.
Can I bounce two things off you before we go a little further? Because for
We need
those
step, remember there's step 4. We're
I
not
know.
there yet.
Before we come on to step 4, just unpack a few things here because we've got this amyloid beta and the
Yep.
p-tau, and there's a lot of energy right now going into a blood test looking at amyloid beta and p-tau
Indeed.
217.
Indeed.
Now, at the
Yep.
conference that you spoke at so brilliantly for Food for the Brain, the Alzheimer's Prevention New Frontiers, one of the extraordinary presentations by Professor Jonathan Stone showed brain scans where you can identify exactly where bleeds have occurred. And literally in any demented brain, there are going to be about 1,000 or more spots, tiny evidence of bleeds, micro bleeds. You may not have even noticed. We're not even talking mini strokes here.
Yep.
And then in the next slide, he showed there's a way of scanning the brain where you can show where the amyloid beta is.
Yep.
And it's identical. It's
Yes.
just the same place. And this
Indeed.
professor of biology asked the question, why are we evolutionarily designed to produce amyloid? I can understand this idea that now we will make more through the process of our bad diet. And the implication there was that we actually produce amyloid in response to a bleed. It's
Indeed.
what protects the electrical system. How does that
It's
tie
like,
into
it's
what
like
you're
sticky
saying?
tape. It's
Yeah,
like sticky tape.
yeah.
Sticky
And
tape for the brain.
and
So
you
when you get the bleed, you got to tape the two sides together so that you don't get more bleed.
and of course, if you rip out the sticky tape, the plaster, you bleed, which is the
Yeah,
biggest, most common side
indeed,
effect of these drugs. But are you
indeed.
saying that
So,
the process of our, SAD diet, as we call it, standard American diet,
yep.
is generating more amyloid? Number one. Yeah.
Well, it is generating more amyloid, but even more importantly, it's being cleaved and
Hmm.
cut in such a way as to generate A beta rather than
Hmm.
uh, products that are beneficial to the cell. This is the Dale Bredesen, connection versus regression, concept. Okay, but it's ATP dependent. It's dependent on the energy inside the cell. That's the primary driver, as opposed to, other things that have been promulgated.
And
We
before
think this is an ATP problem.
Yeah, and before moving on to step 4, the other thing I wanted to say is we know very clearly that raised homocysteine increases and generates p-tau. There's two enzymes, one which makes p-tau and the other which stops it. And we should be clearing it from the brain. And we know that raised homocysteine leads to more p-tau, but we also know, and we're talking here about B vitamins, and particularly
Right?
B12, folate, and so on. And a lot of people don't think of the
Well,
B
think about it
vitamins.
this way.
Yeah,
Think about homocysteine as the
yeah.
byproduct of a defective Krebs cycle.
Well, I was going to say that, when homocysteine goes up, oxidation goes up. And those B
Yeah,
vitamins are not just cofactors in metabolism. In other words, if the energy factor isn't working, you've got this issue, but also they are directly antioxidants. So
well,
homocysteine leads to
indeed,
oxidation. That's the next
indeed,
part of this. Yeah, it all
indeed. So,
ties in
so,
very, very well. Yeah.
so homocysteine is both a product and a cause. It's,
Hmm.
it's a toxic intermediate, but it's generated by the ATP depletion.
Hmm. Yeah.
That's step 1, 2, and 3. Now, step 4, the microglia. And this is where David Perlmutter's work comes in. Okay. So microglia exist in two forms, M2 and M1. M2 is basically scanning for trouble. M1, it found the trouble.
And
Okay.
just go back and explain what the glial cells are doing. You spoke about astrocytes before. What are the
Right. So
glial cells doing?
astrocytes nourish the brain. Microglia basically are the immune system of the brain. They're patrolling, looking for trouble. They're looking for foreign invaders. They're looking for damage that has to be repaired, cleaned out and repaired. They're basically macrophages that have to be activated in order to do their job. They're sitting in wait, okay, and they have to get a signal to basically, become become active participants in the cleanup process.
So they're the
Okay,
brain's defenders, which is the
they are
title
the,
of
and
Dr. David Perlmutter's new book. Yeah.
that's correct. That's correct. And I learned all of this from David, in fact. So the M2 macrophage has to become the M1 macrophage. Well, what does that? What turns a microglial cell on? Answer, ROSs.
So we're talking about going from a brain defender to a brain attacker.
Yes.
And,
And
what makes
ROSs
that
are the
happen is the ROSs.
ROSs because they activate NF-kappa B. And NF-kappa B is the intermediate that drives the inflammatory process and turns M2 into M1 microglia, which basically are like macrophages. And so what they're going to do is they're going to clear out any neuron that is dysfunctional. And if you've got p-tau 217, because you have glycogen, or if you have A beta because you couldn't fold it right, that microglia is going to clear it out. And so you're going to lose neurons. That's called Alzheimer's.
And that's step 4.
That's step 4. You need all 4 steps. You can have problems with step 1, you can have problems with step 2, you can even have problems with step 3 or step 4 separately, but it's the combination of the entire kit and caboodle, all 4 steps that actually gets you to Alzheimer's. And this is one of the reasons why you can see plaques in normal people, like the nuns study, is because you have step 3 without steps 1 or 2, or you can have, step 4 without the other problems, which, can occur due to, say, an infection,
We're
because
talking here,
infection
like, for example, a herpes infection. It triggers
or
the immune
or
system in the brain. The microglial cells go a bit
or
crazy.
or porphyromonas gingivalis
Yeah, from your teeth,
from the mouth.
poor dental hygiene. Yeah.
Okay, so you can have from a traumatic brain injury or so you don't necessarily have to have all 4 to get damage, but when you have all 4, that's called Alzheimer's.
And what is very beautiful about your hypothesis here is it's such a good illustration of what we call systems-based medicine. That our biology is complex, interactive systems. Of course, if you want to make money in pharmacology, you want reductionism. You want one drug or one treatment that blocks, one process. And the
Right.
further we dig into Alzheimer's, dementia, but many things, we are really learning that there are many parts that have to work together to stay healthy. And you're putting ROSs, reactive oxygen species, either endogenous, produced inside you, or exogenous from outside, at the very central of a breakdown, really.
I believe so. That's what the data say to me, and we're writing it up now, very specifically, with that in mind. And I will, David is Perlmutter is one of the co-authors of this, and this has been vetted with him. But there are many other people on this. Perhaps you're familiar with George Perry.
Mm-hmm.
Uh, he is one of the people who exposed the Alzheimer, the amyloid scam
Mm-hmm.
way back when. So he's a co-author on this as well. So we're very excited about this, and it makes sense. But what it says is 2 things. Number one, uh, Alzheimer's is not going to be medicatable
Mm-hmm.
because there are too many steps. And in fact, we've tried to medicate it, and it hasn't worked. And if you look at the few drugs that have actually made it through, they're not really stopping the process. They're slowing it, but they're not stopping it. So I don't, based on this 4-step uh, process, I don't foresee that we're going to get a drug that's going to fix this. So that's the first thing. And then the second thing is that so many of the ROS generators are things we can't do anything about. We're just going to have to live with them. But the one we can do something about is food. Fructose being the worst ROS generator of all, but you know, uh, 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 what do you call omega-6s generate ROSs, especially when you heat them. Trans fats. Virtually everything in ultra-processed food is an ROS generator. And I think this is one of the reasons why ultra-processed food is, sort of the worst possible and mediator of Alzheimer's disease. But what that also says is that we have a chance for prevention because all we have to do is get the ROS burden down. And how do you do that? Well, you eat foods that are high in omega-3s, low in omega-6s, haven't been heated, okay, don't have fructose, okay, and have lots of micronutrients. That's called real food.
Mm. And of course, nature produces fructose. So what's the
Well, yes, but
difference?
fructose produced by nature also comes with its inherent fiber. And when
Hmm.
you consume fructose with fiber, which is called fruit, you actually didn't get the fructose. Your microbiome did, because the fiber acts as a gel, as a barrier on the inside of your duodenum, preventing glucose and fructose absorption into the bloodstream so that the liver never sees it. Reduces the glucose response, reduces the insulin response, thus maintaining insulin sensitivity, keeping your carbohydrate exposure low, and delivering that carbohydrate to the microbiome where it can chew it up for its own purposes. So even though you ate it, you didn't get it.
So eat your fruit, don't drink it.
Eat your fruit, don't drink it, and eat your fiber
Hmm.
because the fiber is not food for you, it's food for your microbiome, it's food for your bacteria. And if you don't feed your microbiome will feed on you. And to that end, and a shameless plug, I am the chief medical officer and co-founder of a fiber company called BioLumen. And BioLumen that makes a product called Munch Munch. And we are actually launching in the UK in June 2026 at the Integrative and Personalized Medicine Conference.
And allulose, where does that fit in all this?
Well, allulose is an epimer of fructose, but it is not used for energy. It does not enter cells. It enters the bloodstream, but goes out in the urine. It has a mild sweetness. It's got a sweetness index of 70 compared to sucrose, which is 100. So it's a sugar extender. And we are using it here in the United States for chocolate, and we're advocating for it to be approved by the EFSA and the Gulf Cooperation Council so it can be brought to Europe and the Middle East. And I am working with a food company in Kuwait called the Kuwaiti Danish Dairy Company to basically take the sugar out of their diet and substitute it with allulose.
And of course, on the one hand, we're trying to generate less ROSs.
Correct.
On the other hand, our metabolism has this exquisite mechanism, well, it's a complex mechanism for disarming these oxidants. And very central in the blood is vitamin C, which becomes oxidized, is then recycled by glutathione, which becomes oxidized, becomes GSSG. That itself is then recycled by niacin in essence, NAD, and also coenzyme Q. So I've, being, approaching 70, I've wanted to absolutely make sure that I've got as little input of oxidation and as much ability to disarm the oxidants. And then, as you know, we developed the test, which is the ratio between the fully loaded glutathione and the spent glutathione, the glutathione index. And I'm just
Well,
monitoring that because I want to keep it low. And by the way, our initial research, which is on a very small number of people, show very clearly a decrease in that increase in that glutathione index in healthy people and a decrease with age. In other words, those factories, those mitochondria, tend to be less efficient with age. So the older you are, the more you need to absolutely control your input of reactive oxygen species and the foods that you eat that generate them, like the ultra-processed foods, and potentially increase your intake of antioxidants.
indeed. And let me make one more comment. You know, everyone thinks that Alzheimer's is genetic because of ApoE3, ApoE4, sorry. Um, it turns out ApoE4, when you actually look at the molecule itself, has to basically transfer electrons. And because of the structure, the change in the shape of the protein molecule of ApoE4, it increases the distance that the electrons have to traverse when they're transferred. And what that does is that generates a lot of ROSs.
Mhm.
So in fact, ApoE4 is an ROS generator.
Right.
So probably why ApoE4 is a driver of Alzheimer's, not because of the gene itself, but because of the ROSs the gene makes, the protein makes.
So you got to work harder if you're ApoE4. Get those ROS input down, get your defense system up.
Indeed.
Yeah. Well, Rob, that is a really interesting and fascinating and deep and meaningful conversation. It feels a bit like Metabolical, the sequel to Metabolical, a whole nother layer. I
In
don't
a
know.
way,
Are you going to write a book on this or are you?
well, a paper first.
Yeah.
First a paper, then we'll see.
All right. Well, thank you very much for your sharp intellect diving deeply into. We've been talking about the generation of dementia, but let's be frank, this is a mechanism which can explain so many of the 21st century diseases that we have
Indeed.
created. So, thank you for adding this extra and very, very important layer.
I offer it to the public and let's, hear the feedback.
Thank you very much, Rob.
My pleasure.